I just want to say thank you for the invite. NCITA, I think, is just a remarkable network that you're building there. And it's been wonderful to see how things are changing and how things are growing and developing. One of the things I was asked to talk about was basically PET and annotating cancer with PET. I took a little license. I'm also going to expand this into some of the areas, I think, in theranostics, where PET and theranostics are intimately associated. And so over the next, I think, 30 minutes, I'm just going to give you some examples of where we are, but where I think we need to go. And hopefully a lot of this technology will be appearing more in the UK in the future. Disclosures, I put them all down, really nothing relevant for this talk. It's just I list them all, I suppose, me doing it individually for each talk. So I'm obviously in the business of radiopharmaceuticals. Small molecules, antibodies, peptides, nanoparticles, viruses, adenoviruses, so on and so forth. We had the opportunity to turn all kinds of wonderful platforms into remarkably good imaging and theranostic agents. I'm not married to any single one of these. When I was a graduate student back in UK for Phil Blower, I was a small molecule guy. Then I came over here and I started doing peptides and I did antibodies. And now I'll just do whatever works and for whatever purpose, because everything that we do always has to have that fundamental clinical question at the end. What are we trying to see? What are we trying to treat? And everything we do on the development side here has to have a goal. We spend a lot of time making agents that we think are going to be great for looking at process A or something about this particular cancer. And then the oncologist's like, well, I don't need something like that. These patients live 50 years. What I need is something for this. So we look at the end and then we work backwards. You know, what is that clinical question that needs a tool for? And then we hopefully build it. And any one of these platforms might be the most appropriate thing to do it. So at MSK, we have taken this approach. And so we have all of these radiopharmaceuticals. FDG is the main one, of course, still. But we've developed a whole suite of small molecules, peptides, antibodies for the imaging and diagnosis of cancer. All these are research, clinical research at this point, looking at things like the androgen receptor, estrogen receptor, expression of PSMA. Everybody knows about the approval of PSMA right now. Antibodies, where we're looking at all kinds of things like HER2, so on and so forth. I'm going to focus really on the antibody portion today, but the reason for showing this is the fact that that's not all you can do. There are these small molecules and peptides that we can access. So when it comes to antibodies, again, what I'm going to focus on today is the fact that these are incredibly specific entities that bind. Sorry, can you hear me? I heard a question in. No? Okay. So we had these antibodies, they're specific, they have great affinity for their target, they're stable, and they give you really interesting and important information about biology. There are obstacles to their use. The immunogenicity. They are slow in their pharmacokinetics. For anybody, I know you don't all spend all day and night reading my papers, but if anybody knows that one of the biggest disadvantages of antibodies is they're so pharmacokinetics, and we do click chemistry to overcome that, and click chemistry won the Nobel Prize today. So I have a whole bunch of interviews today, and it's probably we kind of validated what we do, the fact that Carolyn Bertozzi and Sharpless actually won the Nobel Prize for this today, but that's not what I'm talking about today. We have to marry a radionuclide with that delivery platform. So fluorine 18 is the basis of FDG, but it has a radioactive half-life of 2 hours. We need to have a nuclide that we can match with an antibody so that when we're imaging that antibody and it's taking days or weeks to do its distribution, 2 hours isn't going to cut it. So zirconium-89 has become our isotope of choice, has a 78-hour half-life, so we're able to image at least a week the biodistribution of an antibody. The other advantages are it's cheap, it's easy to get, it's commercially available. When we were doing it, we made it in the US, the only place, but now there's like 4 or 5 places you can purchase it from. And it's the basis, I think, of at least 10 phase 1, phase 2, and even a phase 3 trial right now. So in the last 10 years, the availability of this nuclide has really just shot through the roof. And now you can get it, and it's pretty cheap and easy to use. We started this again with that clinical question. And the clinical question that came up, and whoever's heard me talk seen me give this particular story many times, but I think it really is a perfect example of what we're doing. Yelena Janjigin, she is our chief of our GI service, about 55 medical oncologists that she has in her team. And their goal is obviously pancreatic cancer, GI cancers, gastric, and so forth. Her particular interest is in gastric cancer. And she came to us wanting to see if there's a way that we could delineate the third of those gastric cancers which are HER2 positive. A lot of work was done into this, but needless to say, we already have a phenomenal entity for targeting HER2, namely Herceptin, trastuzumab. And we can easily take this antibody, label it stably with zirconium-89, and start interrogating gastric cancers for HER2 status. In mice, where we would call start everything, we can take gastric cancer, positive and negative for HER2, bilaterally implanted into a mouse. We can look at FDG, no difference between the two tumors, FLT for proliferation, no difference between the two tumors. But when we give the same mice Herceptin labeled with the PET isotope zirconium-89, obviously the mouse on the left is the one that's HER2 positive. So we know that we have an ability already non-invasively to discriminate HER2 positive gastric cancers. If we then take patients who are getting afatinib as part of their therapy, one of the most important things is the third bullet, that if a patient is gastric cancer, has HER2 positivity, and is getting afatinib, that it inhibits the HER2 receptor. So we can see if afatinib is working in a particular tumor based on the readout of the HER2 expression in that gastric cancer. In mice, getting a dosing of afatinib that a patient would get, you can see that FDG doesn't change over a 3-week period. But in the same mice, if we interrogate for HER2, baseline, great uptake, but then it drops off immediately because the afatinib is working this tumor, therefore it's downgrading the expression of the HER2. Because we're taking an antibody that is already in humans, HER2, and it's given at huge doses of 3 mg per kg for days, and men and women have been on this for a long time, we have the ability just to take that same antibody and label it with zirconium and put it into patients without any additional toxicology packages per the FDA. Because we're not giving it a 50 mg per kg, we're giving it 3 mg total to a patient. We're at a microdosing level. All we're giving is 3 milligrams total body. And so when we put this into patients with gastric cancer, this is obviously the central view you're looking at here, we can take a patient at baseline, then 21 days, re-image them with zirconium trastuzumab, and then 116 days. And this is one example that I think really shows you the power of imaging. And that's we have these 3 lesions lit up by the white arrow with the black surround, the black arrow, and the white arrow. This patient, gastric cancer, male, getting afatinib daily. But look what happens to the tumor that has the white arrow with the black surround. It disappears. Maybe not volume-wise, but the HER2 signal has disappeared because the afatinib is actually treating that tumor. The tumor, however, with the black arrow at 0 and then 21 days doesn't change. So the afatinib is not working in that tumor, and then it continues to grow and actually kills the patient. So we can look at an intralesional heterogeneity here. Oh, and the third tumor, part of it responds, part of it doesn't. But by a HER2 readout on an individual lesion basis, we can say which one's going to respond, which one's not. We know because that signal disappeared at 21 days, that one up, the higher up one, it's going to respond, and it did. When the patient went off to autopsy after the study, there was no tumor left. That one where the signal didn't change continued to grow. When we looked at the pathology of that tumor post-mortem, again, it completely confirmed afatinib wasn't going to work in that tumor. So we can, in a individual patient basis, look at mixed response. And maybe that tumor that wasn't responding, we knew after just 21 days that that tumor wasn't going to respond. Maybe a different drug or a surgery or something else could be considered in the example of this patient. Now, of course, PD-1 trials are hugely important right now. So we're using this HER2 imaging in that context as well, where in a trial where the patients are getting pembo and trastuzumab, and then pembo, trastuzumab, chemo, we're using, as you can see in these 2 images, the readout of HER2 as a way to see how well these lesions in these patients are responding, even on mixed trials, by using HER2 as a readout for the effectiveness of the therapy. So with that patient I showed you, we had these mixed responses. Now, somebody could argue, well, we could biopsy it. But you know, when you're taking a biopsy, you're taking a very small sample from a very heterogeneous population. This paper I get everybody to read because it shows you in this primary tumor, they did 9 biopsies and they had 9 completely different gene signatures in this primary tumor. So if you're taking one biopsy from this and basing your entire patient treatment on this tumor, you're not going to have a good response because the heterogeneity is present. But they will always be important, and we will always do biopsies, but we can also do them informed. If we can actually get the right images, we can tell the interventional radiologists which tumors to specifically biopsy. So we can start doing informed image biopsies based on not just about the presence of a tumor, but something about the biology of the tumor, like go in and get this one. With serum biomarkers, you have the opportunity, obviously, something very cheap, that whether it be blood or sputum or feces or whatever, you know, these are phenomenally important, particularly in developing countries. And I have my PSA test every year now because I got to that age. But, you know, it tells you something's there, but it doesn't tell you the source of where that biomarker is coming from. So if you have a marker that's absolutely perfect for saying you have tumor, you still have to find out where that tumor is coming from. If you're using it as a marker to look at response, and that patient I showed earlier that has a mixed response, and you're relying on a blood biomarker, it might just be averaging the response of 2 tumors. That tumor continued to grow, but one tumor shrunk. But if you relied on a biomarker from the blood, it may just say that there's stable disease. It's not showing you anything else, but you still need imaging. With imaging, of course, we can do it non-invasive. And if we start marrying these serum biomarkers with the tissue biopsy and the imaging, doing informed imaging, informed biopsy, we're going to get a much better idea what's going on with these patients. And that is an area, I think, that particularly with the technologies going on in the UK, that the biomarker programs, and you know, Karen and I's work, and Charlie, somebody else's, that the opportunity to start marrying these biomarkers with imaging is something we really have to get more into. One example of that is in the CA19-9 world. This is obviously used in many cases of PDAC. It's the Lewis antigen, not named after me, obviously. And it's something that's used as a serum biomarker for patient with pancreatic cancer, responses to therapy, so on and so forth. We had an antibody, 5B1, that was isolated from a patient. So it wasn't humanized or made in a test tube. It came from a human being. So a human being made this antibody, but it had incredible, exquisite sensitivity or selectivity for CA19-9. So we have a serum, and it's used as a serum biomarker, the CA19-9. So we now have a tool to find the source of that serum biomarker. So using mouse models of cancer, obviously orthotopic in the place of pancreatic cancer, we can take this 5B1 antibody that looks at CA19-9, and we can find the source of where it's coming from. And in this case, on the right-hand side, it was from a orthotopically implanted pancreatic cancer in the pancreas of a mouse. Obviously, the FDG on the left-hand side, FDG isn't great in the pancreas cancer world, so that's why we want something better. When this goes to other models of PDAC models, you can see again how beautifully it delineates these pancreatic cancers, which express CA19-9. The CA19-9 is obviously a shared antigen because that's how we can detect it in the blood. But what we're doing here is finding the source of it, and the source is in a higher concentration. So that is why we're able to see the tumor. When we go to the patients with this, first of all, it shows you how it takes 5 days to get the images we want. But this is a pancreatic cancer patient who has CA19-9 positive disease, as shown by serum biomarker. But what we've done here is injected this antibody, and we can see by 5 days where that CA19-9 biomarker is coming from. It's coming obviously from a lot of liver metastasis present in the liver, as well as even thoracic node up there. And if you're relying on the serum biomarker to be a measurement of response, is it one of these tumors responding, all of them averaging? But because we can now visualize the presence and location of the CA19-9, we believe that this will also be able to give us the ability to do an interlesional, intralesional, individualized readout of which tumors are responding and which ones aren't to a given therapy. So we've extended this into many other cancers. We've taken daratumumab, which is used in multiple myeloma. On the bottom there, you can see how well it lights up the tumors of this patient with multiple myeloma compared with FDG. Also pertuzumab I've shown on the right-hand corner. I wanted to show pertuzumab and the fact that it also delineates and lights up brain metastasis. People like, well, antibodies don't get into the brain, but when there's a metastasis there, the blood-brain barrier is broken down, and it really does light them up very well because there's no background. And we're seeing more and more brain mets, and say even pancreatic cancer, because patients are living longer and these mets are now presenting. So we always have to make sure that we can see some more difficult things for the oncologist to deal with. And in this case, brain mets really do light up extremely well, at least in all the patients we've done when it comes to imaging and antibody. But if we get this kind of image and we're able to show these kind of results and this kind of uptake, if we're able to delineate this and get this as an imaging agent, why don't we take off that isotope that's used for imaging and put on an isotope that's for therapeutic? And that is where the world of theranostics comes in. See what you treat and treat what you see. So if this patient here had no uptake, don't give the theranostic therapy because there will be no uptake, no damage. But in this patient here, obviously, you could argue that a radio-labeled antibody with a therapeutic payload would actually have some benefit. And the goal, of course, of radiotheranostics is to enhance tumor therapy, manage adverse events, improve patient outcome, and do it on a more individualized basis. There are many different isotopes that can be used for theranostics, alphas, betas, no J. And each one of them can have different responses based generally on the size of the tumor. But I don't want to, even though I'm going to be talking mainly about betas, there are other therapeutic nuclides out there that can also give good responses in tumors. The concept is this, that you take the patient, you image them, and if their tumor's not positive, they don't have an image and it's negative, then you find a different therapy. You don't go ahead with a therapeutic version of your diagnostic. But if you obviously have something to see and there's some tumor present, then you can start going through with different cycles of therapies, and ultimately you can follow up by more imaging, or sometimes image the therapeutic portion of the antibody. Obviously, this has really come to the fore in the last couple of years. Radium chloride Xofigo was the first real radiotherapy out there that was approved. Well, no, there was Xofigo and Zevalin, but more recently there was Xofigo. Then, of course, along came Lutathera, and then more recently lutetium PSMA, where they're really showing dramatic responses to the therapies. All of these had the ability to image. With Xofigo, you can do bone imaging. With Lutathera, you've got the opportunity to do dotatate. And then you have the gallium PSMA when it comes to Lutathera. Treat what you see and see what you treat. This has been around for a while, and particularly in the pediatric setting. One of the things which I think often we need to spend more time thinking about is pediatrics. We have the largest pediatric volume in the United States at MSK, more than St. Jude's, but we really do have some examples of where these theranostics have worked and profoundly made a difference in patients. So one of these is an antibody system developed by Naikong Chung, along with pediatric neuro-oncologist Kim Kramer. And they were looking at metastatic leptomeningeal neoplasms. It's about 5% of patients, but median survival is less than 6 months, with a 3-year survival less than 10%. So for these pediatric patients, this is devastating, absolutely fatal. But taking an antibody system and using Ommaya systems and having, this was done about 10 years ago now, and patients and using I-131 as a therapeutic isotope, but using, because they're pediatric patients, imaging to decide how much dose they got, so you didn't overdose pediatric patients or underdose pediatric patients. And obviously that helps with long-term side effects, but so you actually gave the child the right amount of therapy that would treat their disease, as opposed to just based on their body weight. They had some results which are just getting reported now, with 80 patients treated, as you know, was probably fatal, but we now have about 30% alive at greater than 60 months. And this is best shown here, that patients under the age of 18 months year old, so very tiny toddlers, less than 18 months old, that there was, when treated with this I-131 8H9, that there was an 80% survival. And if you look at your x-axis here, this is not months, this is years. So these children now, these 18-month-old children, now teenagers and at school, and it's an absolutely universally fatal disease until this kind of theranostic approach came on. And approval is probably going to be this year. And we've had kids fly all over the world for this, because this obviously is quite a remarkable result, but shows you the power of imaging and theranostics, because we can image and do that. We also have the ability with antibodies to interrogate targets which we haven't thought about looking at beforehand. Sometimes in the world of therapeutics, we think, oh, we need so much of the target in order to get the therapy there. That is not the case with radiopharmaceuticals. We can do very small, very low expressed targets. As long as the background is clear, then we can use something like this. In the small cell lung cancer, DLL3, delta-like ligand, is one of those targets. It's only expressed 10 to the 4, but it isn't expressed anywhere else. So it becomes a very good target. It was used as part of an ADC trial, the Rover T trial, that initial results were great, but the phase 3 fundamentally failed. Not because of the antibody is great, it's because they put something so toxic on it that the ADC caused all these side effects, but it doesn't mean the antibody's no good. The antibody's amazing. So we just took this antibody and started looking at mouse models and low-level expressing tissues and found that even the lowest expressing small cell lung cancers, we were able to light up with DLL3. When this was taken to a human, the translation was literally just months because the antibody had already been in humans as an ADC. So we just had the ADC off, so no matter what, would have less toxicity than with the ADC. Turning this into a zirconium agent, you can see that in small cell lung cancer, how beautifully this lights up the liver metastasis from this disease. And we've done this in, this is a 70-year-old patient, but we're seeing this in, we had a 23-year-old patient recently where this was also shown to work extremely well. If we've gone through that trouble of getting this image, let's switch it off. There's a zirconium for diagnostics, turn it into a therapy, and look at the right-hand side that the treating now tumors in mice, you completely flatline, you get complete cures and durable responses in small cell lung cancer by doing this. And the reason is that small cell lung cancer is actually responsive to radiation. Unlike some which are radio resistant, small cell lung cancer isn't. So we had the ability to get very durable cures over a very long period of time in mice. And obviously our next step for this is humans. If we've gone through all this trouble of making one agent, we don't want to ask one single question with it. And so right now we're facing the issue of neuroendocrine prostate cancer, NEPC, because when we're treating patients with prostate cancer, which are AR positive, PSA positive, PSMA positive patients, and we're giving them lutetium PSMA and getting these great results, over a period of time, the tumors which aren't responding, or small cells which aren't responding, are going through a treatment-induced transdifferentiation. So now these patients come back with prostate cancer, that's neuroendocrine prostate cancer, that has no AR, no PSA, no PSMA. And once this is present in the gentleman, it's incredibly aggressive. It is resistant to hormone therapy. And once you have neuroendocrine prostate cancer, overall survival is less than 1 year. Fortunately, and this is going to be a growing problem. So if you look at the incidence of neuroendocrine prostate cancer from 98 to 11, it was 6%, but from 12 to 16, it's 13% because PSMA started coming in. So now that's approved, this number of 13% is going to dramatically grow because we're just going to have all these gentlemen now with neuroendocrine type prostate cancer that have responded to PSMA and now they're failing. And the one advantage of neuroendocrine prostate cancer is the fact it expresses DLL3, that target that we just hit on small cell lung cancer. So we already have an antibody in humans that's used to interrogate delta-like ligand 3 in small cell lung cancer. All we have to do is change the protocol, say, let's do prostate as well. And so we did that in mouse models of cancer. We imaged mice that were neuroendocrine prostate cancer that came from PDXs from our collaborator Yu Chen. We imaged them with PSMA. The tumors were negative. We knew they were going to be. We imaged them with DOTA-take because we thought we'd give it a try. And then we also imaged them with SC16. And this beautifully lit up the neuroendocrine prostate cancers in mice. We were able to recruit patients immediately. And here's one example. It's an interesting example, but it's an important one in the fact that on the right-hand side, you see the FDG PET of all of this patient who has advanced metastatic prostate cancer. Everything lights up. On the left-hand side, the same patient got SC16, but only 2 lesions lit up. And that's actually okay, because once you have the neuroendocrine prostate cancer, your overall survival is less than a year. So these are the 2 lesions which we're going to have to watch out of everything else. But we know he has this now, so we have to do something actionable against those. But right now, there's nothing approved for treatment or imaging of neuroendocrine prostate cancer, but there's at least SC16. And just we did in small cell lung cancer, if you then take these tumors and you treat them with lutetium version of the antibody, again, we get cures and incredibly durable responses in these mice. So we believe that this guy would also respond beautifully lutetium SE16, because again, neuroendocrine prostate cancer is actually sensitive to radiation. If we don't have that ready, then maybe the radiation oncologists need to go through and just zap those 2 lesions, because they should respond, and we know exactly where they are now. And of course, we can also confirm that by telling the interventional radiologist, biopsy those 2 and confirm it's neuroendocrine prostate cancer. So where's the field going? And this is why I'm just wrapping up the last 2 slides in the last 3 minutes. A lot of these therapies with lutetium have been done with betas. Alpha radiation is the hottest thing right now. Using alphas, and obviously alpha PSMA and alpha antibodies are now in phase 1, phase 2, and phase 3 trials. And it's because the alpha radiation is way more potent, but also it means if you don't get it to the tumor, it can do some also pretty nasty damage other places. So the choice and the sensitivity and the selectivity of the targets are key. But a lot of people are going down alpha radiations now, and this is probably the most overshown slide in history of nuclear medicine, but also shows you so well that a patient who's getting lutetium PSMA failed 2 treatments, and as soon as this patient, this is all metastatic prostate cancer, all that black that you see, that he failed 2 treatments of lutetium, which is now the FDA approved drug, but when he was given the alpha emitting PSMA, you can see how well it completely destroys all of his pancreatic cancer, prostate cancer, the fact the image on the right with the 3rd cycle, he now has undetectable PSMA. Not without side effects. You can see the salivary glands lit up there. And so xerostomia is an issue, but there are new analogs coming out. But this really does show you, I think, the power where alphas can come in after beta if somebody's failed a beta therapy. Is this, we just finished this article here, is this going to happen? Yes, it is, because the market is driving it. In the US, we've had hundreds of millions of dollars being plowed into radiopharmaceuticals right now, and startup companies. And as you can see from 2013, when Xofigo was approved, that with this market by about 2025, we predict it's going to be at least about 10 billion a year. It's going to be huge, and it's going to continue growing. So the incorporation of theranostics into all aspects of radiology, into medicine, into treatment for cancer is absolutely going to continue to grow. And if departments aren't ready to do radiotherapeutics, other departments will, and other places will take over. It also allows you to also hit up a huge number of new targets, which you've never explored. I think Eric Abouaji is on the line, and I think he would look at this and hopefully agree that there's a whole bunch of other stuff we have yet to hit. I know that there are some plans for some new centers going on in the UK, where new targets will be identified and isolated. It should come with a concomitant design of probes and therapeutics to it, so we can both image and treat using this plethora of really amazing targets which are coming down the pipeline. Finally, I talked a little about the lab work, but this is my current lab, and I can't always sit here and without at least acknowledging them. This group is the key group in terms of translation. This is our radiopharmacists and radiochemists who get all of this stuff that I said was translated. They're the ones that truly make this happen. I just had about 30 seconds left, so I say thank you very much indeed. I hope you're able to hear that. If not, then it's been a horrible disaster the last 30 minutes, and I know I rushed through things pretty quickly, but if there's time, I would be delighted to answer any questions. Thank you.